Hi everyone, welcome to today's seminar. I'm Malcolm Macaulay, one of the co-organisers of the IBH seminar series, and before I hand over to Luke Ramsey, who's going to host today's talk, I just wanted to let you know there's a couple of options for asking questions. You can either type your questions into the chat and Luke will ask Isabelle your questions after the talk, or after the talk, you can virtually raise your hand during the Q&A session and you'll be unmuted to ask your own question. So thanks, enjoy the talk, and now I'll hand over to Luke to introduce Isabelle.
Hello, it's my great pleasure to introduce Isabelle Collard to give today's seminar. Isabelle hails from France and started off working for industry in a company called Agrogène, who was a specialist genotyping company for the breeding industry run by Pete Isaac. But she saw the light and she moved to the UK and did her PhD with Graham Moore and Peter Shaw at the John Innes. We're working on wheat, but then she really came to her senses and came up to Dundee to start to work on barley. So this is a specialist presentation on the specifics of barley meiosis. Isabelle, the floor is yours.
Hello, everyone, and thank you very much for inviting me to give this seminar, and thank you for the organizer and everybody who actually can make it. Today I'm talking about meiosis and recombination, but I really want you to think about dancing. So we're going to dance today. So first of all, I'm going to try to find where to move in my slide. Here we go. So I'm going to give a brief introduction about why barley. So I think I got the blue screen coming up, so it's not very good. Okay, so this is barley is important. I think I don't have to say anything to this audience, but it's the fourth most cultivated cereal grain in the world. But a lot actually goes to feed the animal as well. So I think it's something that you need to think about very carefully here. This is a staple food in high altitude areas. So for example, I know that in Peru, they have the barley, in Ethiopia, in the highlands, and then also in Tibetan Plateau and Himalaya. In Scotland, we like it for whiskey. And there's a very, very massive economy for us. So for me, barley is an excellent model for large genome cereal research. But I want to bring your attention on the very, very big size of the barley genome. So it's twice the human genome. It's got only 7 chromosomes, double 2, because it's a diploid. So it's a 14 large chromosome compared to human, who has 23 for a much smaller genome. And of course, it's much larger than Arabidopsis, which is the model plant for geneticists. But it's a lot smaller than wheat, which is then a lot easier to work on. And like many other crops, it's facing issues like extreme weather, heat stress, grain feeding. The grain quality is sometimes not up to scratch, and it needs a lot of input for low nitrogen use efficiency, and this is at break. So we still need better variety. And this is why we're doing this work. Because in conventional breeding, the main way to get an extreme good variety is by recombination. And here I'm going to explain why it's not optimum here. So if you look at this graph, on this side here, you have the total number of crossing over for the whole population. And at the bottom axis, you have the approximate location on the chromosome. If you were to plot the physical chromosome to this graph, you will see that actually most of the recombination is happening at the end of the chromosome. But it doesn't mean that there's no gene in the middle. It just means you can't access it very easily. So for us, the game is to try to break that up. The second issue we have in barley breeding is the recombination in the hybrids. So here's an example. If you imagine a block, this is your region of interest. In green, you have the allele that you want and keep. And in blue is the one that you really don't want for whatever reason. So if you have only one recombination event at this position, and then another one very far away, you're basically moving the whole block containing both beneficial alleles and deleterious alleles. But if you increase recombination events, then you can start to break that up. and to break the pattern and select the variety for what you really want. So that's another thing we need to try to find out how to break that up. So in summary, right now, we want to be able to modulate recombination by increasing recombination, but also by shifting recombination in the centromere area of the genome. So that's where we are. Let's dance. I call it the meiotic waltz. So first of all, the mom and dad goes into the ballroom and they start to shake hands. They need to find each other, those chromosomes. So in blue, you have one parent, and in red here, you have the other parent. The first movement is called synapsis. So that's the first step of your dance move here. And what is happening is that each of the homologous chromosomes needs to find each other and align along the entire length. It seems easy, but you will soon realize that it's not easy at all. And then you have the crossing over movement. This is where you need your recombination. This is where you need, you have gene exchange. And they literally break apart to exchange chromosome from one parent to the other. The next movement is a swap, by meaning of each of the parental chromosome homologous goes to each side of the nucleus and get divided for a second round of division. That's giving you the grand finale with the shuffle mixed. And that's each of the haploid cell here has a very unique individual genetic makeup. So we're going to dress up for synapsis now. So how you dress up for synapsis? Like I said, it's a very complex mechanism, but you can track it down by using antibody raised against specific proteins. So here is a representation of what we call the synaptonode complex. This is a tripartite structure that is formed to help the chromosome to pair and align along each other's. So in green here, there's a protein called ASY1, which is normally coating the chromosome axis here and organize the chromatin loop for each of the parental line, parent chromosome. In the middle, it's being paired up or zip, like a zipper-like gene called ZIP1. And in all my talk, ASY1 should be in green and ZIP1 will be in magenta, so you can track it down. So this is how meiosis will look like in Barley in real life. It takes about 48 hours in Barley to run through meiosis, and more than 33 hours comes into all this area here up to metaphase. So the half top, it takes 2/3 of that time. And there's a very good reason for it, is because synapsis is happening and recombination is happening. So the first step is to find your partner, to go into the waltz. To find your partner, the protein ZIP1 is taking the chromosome from the end of the telomeres, and then they start to zip up from the entire length. So by a stage called zygote, you normally have the pairing up of those chromosomes along each other. By the stage where we call pachytene, the chromosomes are fully paired, but then they start to twist and twirl. It's a waltz after all. Then they start to dissolve, and they take again another remodeling for that. So this dynamic is very important for us, because when you understand how it should look like, you can easily detect what doesn't work very well. During metaphase, we call them chromosome bivalent or donuts, because sometimes when you dance, you need a little bit of refreshment. And the representation is not accurate in here, but the essential idea is that this is the chromosome of the first parent, the chromosome of the second parent. Each chromatin is attached by the centromeres, and then the telomeres, are they attached together, so we can link the bivalent by crossover. So that's basically how it will look like on the picture. And then they need to separate. So here you said, okay, waltz is finished. Parent 1 goes to one side and the parent 2 goes to the other side. This is the first meiotic division. It's dividing the diploidy, it's dividing the chromosomes to end up with 4 haploid cells after a normal mitotic division. And if you remember, each of them has a very different genetic makeup. Right, let's go for the great audition. Then we need dancers, we need to select them. How do you select them? So we have a huge collection of mutants that we are calling the Bowman line, which have been identified in 70s, backcrossed many times in a common cultivar called Bowman to create near isogenic line. And I've been fortunate to have like 14 mutants called desynaptic mutants for potential defect in meiosis. So they are spontaneous mutants, they are at the top. We also have produced ourselves mutant by RNA technology or by CRISPR. So here's an example of a mutant called FunkHem, which normally is very good for increasing crossing over, but probably not in body. And we also have a massive collection of teething mutations. So we probably have at least 2 or 3 collections now in the Hatan. I started with the optic mutant, which is a very small collection, but very useful for me. We develop a teething called in Golden Promise. It's highly mutagenized, but it's still very valid. And I think Katie has talked many times already about the collection of laureates that she's developed in the recent years. But today, I'm going to really focus on my favorite desynaptic mutant. So here's the unbalanced ensemble. Here we go. Here are the dancers today. So how you analyze a metaphase behavior? So here on the side, this is the wild type situation. You have 1, 2, 3, 4, 5, 6, 7. 7 ring bivalent, perfectly aligned on the metaphase plates. The telomers are there, and they are pulled apart by the centromeres on each side. They're already starting to be ready. But other mutants are not really dancing very well. They're a little bit offbeat. Some of them don't form anything at all. So what's going on? So 1, 2, 3, turn, 2, 3, 4, swap, 2, 3, 4, cross your arms. That's basically meiotic recombination, right? So that was my very, very simple representation of recombination. But in reality, you imagine this is far more complex. This is what is happening at the DNA level. You need to start by breaking the chromosome. That sounds counterintuitive, but that's very important because you need to repair. Then you need to resect so you can have this strand getting invaded. So if this homologs doesn't invade this other homologs, there's no recombination possible at all. It's just going to be a normal DNA repair. There is something called double holiday junction that start to secure this strand invasion, and it start to repair. And then those little structure we call D-loop, they start to move across along until they finally resolve into a crossover or non-crossover. Now, just for the record, there's maybe about 700 breaks made in the genome, but there will be only 20 or 21 crossover. That's because this is a heavily genetically controlled. So let's call about the wallflower mutant. Wallflower people normally, they're a bit alone, they're a bit shy, and they dance alone, right? But that's a bit the case for DMC1. He's a bit alone here. And what is happening? So DES5 is one of the desynaptic mutants that has a mutation in a gene called DMC1. And DMC1 is known to be working and promoting strand invasion for promoting crossover. So what will happen? Well, first of all, you can see that it's not going very well because the plant is nearly sterile. We get about 5 or 6 seeds per generation. This is a nightmare to work with, but we've managed to make crosses anyway. So well done us. This is a 12 base pair insertion as the it's just at the junction in between the 13 and 14 exon of that gene, and it's basically changing the protein organization. It's changing that catalytic system here, where basically the protein is not being able to work as an exomer. So that's probably what it is. And what it does, it's first of all, you can see that even though This is a gene for recombination. On the top, you have a normal synapsis with the pairing of a chromosome at beginning, and then fully synapsed during piketing, and then a normal dissolution. That looks all very beautiful. But in the mutants, it's very late. In fact, it doesn't really reach piketing at all. It stays in this bicolor type of myocyte and cells because it doesn't actually pair up. If the chromosomes don't pair up, you can imagine a lot of trouble. And indeed, what is happening at the metaphase is chromosomes can't keep together. They are not paired. How should they? They go their own way. They're all free dancing here, right? But the problem is, if you don't have a crossover, you're also losing chromosome in the translation. So you're losing something. So that's not very good. And here's one of the old genetic map that we've done where you can see that at the recombination level, there's very little happening in this mutant. So the take-home message here is the strand invasion leads to abnormal meiosis if it's not normal, and there's no synapsis at all. You have crossover loss, and the crossover loss means sterility. So that's the first take-home message. Let's have a look at the worst done partner ever, right? So this is a gene called XRCC2. And it was actually a very, very nice, promising gene because in Arabidopsis mutants of XRCC2, they increase in recombination by nearly 50%. So it was a very promising target for breeding. Let's have a look. We have two alleles of this gene. We have a very large deletion in BW247, but we have a smaller deletion in that gene in BW248. And you can see that already, as breeding is concerned, it's very unfertile. Very, very unfertile. So you think, this is very different from Arabidopsis, because in Arabidopsis, they are fertile mutants, right? So something must happen here. In terms of synapses, we didn't actually really see a lot of differences. The only differences that we've seen is that during the zygoting stage, there's a lot of loop like this that looks very weird. But basically what it's an indication of a late synapsis. But at the end, it will be synapsing. So it's not a problem here. But at the metaphase, you're losing crossover. That was at least very obvious. So if you're losing crossover, then something must happen in here. So let's go back to the table. What actually XRCC2 is doing? Well, it's playing with his two other friends, DMC1, which you know is very, very important from strand invasion, and ROD51, which is another protein of the family, which is helping also in partnership with DMC1 to help with the strand invasion and securing that strand invasion. So what are they doing? So what we did first is to check that synapsis was indeed very delayed. And for this, we've used something called the EDU labeling. So we've used a modified thymidine that will be incorporated during DNA replications. And then as the cells start to enter meiosis, the cells are divided, and then you will see how they labeled if it divided, if you incorporated the EDU in early replication or late replication, and so on. And what we have as well is custom antibody for ROD51 and DMC1 protein, so we can track them down. And this is what we've done. So here's a couple of images. So the first thing is you can see that, yes, there's a difference of labeling. So the white one here is the EDU. And there's a very difference of labeling in between the two cells. There is almost nearly no labeling at all. That's because the cells were already replicated when they enter meiosis. But for a similar stage of a plant that's been injected with the EDU in the mutant, then the EDU is incorporated because replication is either not finished or is delayed. So that does make sense, right? And all the images were consistent. What we see as well is, if you remember, this time, I will trick you a bit, I dressed ASY1 in magenta. And if you see the localization of the protein DMC1, they start also at the side of the nucleus near the telomere region. But in the mutants, it's much more scarce around. It doesn't seem to be really mobilized in here. On the other side, RY51 changed very little. Maybe the number might be different, but at this stage, it's not really very possible to count them. But the localization is really, really similar. So RY51 doesn't really change his behavior, but DMC1 does. So basically what and of course, because DMC1 doesn't work properly or not in a timely manner, this is the result in recombination. You have the wild type with normal recombination in blue, but on the mutant, you can see that there is very little recombination. And on top of that, everything is pushed distal in the distal part as well. So you're really not getting anything at all and in the middle. So the take-home message here is like Barley XRCC2 is essential for the dynamic of the protein DMC1 and RY51 during stand invasion and stabilization and for the formation of crossover. What if you were coming late to the dance and you were starting to the waltz much, much later in the process? Well, here we have a mutant called MLH3, which is a mismatch repair and contains an endonuclease domain that actually helps to resolve the crossover at the end. And in many organisms, you can have the labeling of this protein nearly at the end of your synapses. Our mutant, as usual, is semi-fertile. And at the time we were doing the pollen staining, and you can see that here you have very healthy pollen with very dark nucleus. But even in the heterozygotes and in the mutant, when they're very sterile, you have a lot of empty pollen or very light shadow pollen, which is probably linked to aneuploidy. But what the most striking things in here for this mutant was that at the time we've used structure elimination microscopy, which is a super resolution microscopy that detects up to 110 nanometer differences in between your foci. And what we've realized that at the beginning of the synapses, again, here's the wild type with the protein ZIP1 in magenta taking up the chromosome together to pair them up. Here's the little cute chromosome pairing up. But the mutant is a little bit disorganized. It still do it, but it's not great. In zygotin, you see the two chromosomes already paired up, and the other half is not paired. So this is why it's bright green, right? In the mutant, yeah, they kind of paired up, but look at the configuration of ZIP1. It's actually a coating on the top. It's not finished to pair up properly. It's not done it yet. And you have all these little loops because everything is delayed. And when you go to packeting, when you have a perfect alignment on the chromosome, here you also have an alignment, but in fact, you have a very spotty synapses. So your synapses is almost complete, but not complete enough to have a very full block along the entire chromosome. And so the result of that will be evident in a minute, because if you remember, I told you that the MLH3 was a protein called a mismatch repair protein. The mutation we have is 159 base pair deletion in a region which is coding for what we call the MUT-LC domain. And that domain is interact with other partners to secure recombinations and events and to secure crossover for resolution. And the wild type here is how the protein we should normally look like, but in the mutant, you can see that already there will be some issues into the conformation of the protein. But the beauty of that mutant is that we still have a protein. And it's still making a protein, but what it does is in the wild type, when synapsis is almost complete, you have almost no signal of MLH3 remaining. The only thing we have remaining is a little foci in here, one here, one here, one here, which is marking the crossing over. But in the mutants, you have a lot of very, very, very small foci, which are not forming a crossover. And you have maybe 7 to 8 foci remaining, marking the actual crossover. And that number is also the number that we found in the total meiotic recombinations in this F2 progeny in the line. So the take-home message here is that the improvisation of inheritance is not very a good dance, right? And MLH mutant is actually one that behaves very much as expected than the other mutant in the world. But what we found here is that the timing was very important when you dance, because what we didn't know is actually MLH3 is not just working in that part of the process. It's already starting here. And for us in Barley, it's very important because that means that the time when you can change crossover is probably happening around this region. So what does it mean? Well, that's a genetic pas de deux, right? You need to really dance together. So what this research told us is that the process of synapses and the process of recombination is very interconnected. And that when you mess it up with one or the other, you either have disastrous comment and disastrous outcome. And here I plotted the gene that we just looked at today, but I want to bring your attention to all the other genes that we found already, and that we need to work more on it. Now, small conclusion, most of the gene leads to abnormal meiosis if you knock out a meiosis or recombination gene. What we already can highlight here is that translating research from Arabidopsis to Barley is not actually very easy sometimes. And it's not straightforward. Even if you think something will work, it's very likely that, well, we don't know actually. It might work or it might not work. We just don't know. And so, can we actually increase recombination in Barley? And actually, is it something that is possible, right? Well, maybe. So let's go for this program, Sticky Come Dancing. And that's featuring the HVST1, the breakdancing prodigy. Right. So for me, again, I just want to highlight what looks like to be a very, very good metaphase with 7 ring bivalent and with a little cartoon to remind you how the bivalent unform. Here are the centromeres. We're putting on each side of the chromosome. And near the telomeres, you have the crossovers going to happen here. But when something goes wrong, you have rod bivalent. So here you have a missing crossover. But this mutant has another things going on. The chromosomes are very sticky. Very, very sticky. And that was very difficult for us to count anything at all. If you wanted to count the crossover and count the metaphase, it was really, really difficult. And we just wonder what was going on. And we were expecting chromosome break and all these things going on. So what did we get? An increased recombination instead. So that was kind of weird for us. But it was also very exciting because this is the first mutant that had an increased recombination despite looking a bit awkward. So that's very exciting for us. So what is AST1? Well, another exciting point here is that this is a single agent gene that contains a ring H2 domain, and this is the structure of the ring H2 domain. So, and here's where this 12 is mutated, right? Or this 4 is the same. And more importantly, we found out that it's mainly in grasses, and we couldn't find anything in Arabidopsis, nor in human. And then we found out that it was annotated as an E3 ubiquitin ligase. And we thought, oh, great, this is a new gene, right? We also find out that it was expressed quite highly in meiosis, and especially in the myocyte. So it's definitely meiosis specific. So that was even more exciting. But the problem in not having anything to relate to and not having any orthologs in another organism, it means that you need to do some more things later on. So let's see. And that thing that we had to show is that that was actually a real E3 ligase. And so for that, we had some advice with people at the University of Dundee to help us to go through this technology. And to tell you what E3 ligase is doing. So basically, D3 ligase is here and is binding with something called E2, which then bind a substrate and add ubiquitination sites on one after the other. So you can have a very long chain or very short chain. And if it doesn't work, then you don't have ubiquitination, right? And on the gel, if you have ubiquitination, you have your molecular mass that will appears higher and appears as a smear. And it's exactly what is happening. So if you mixed an E2 and E1 and ST1, you have an increased molecular weight. But is it ubiquitin? Yes, it is. Here is an anti-ubiquitin antibody, and yes, you still have that pattern. So yes, this is a ubiquitin ligase. More importantly, it's also ubiquitinating a very interesting protein like ASY1. You remember that little protein that keeps on pairing the chromosome and dressing up in green and magenta does this one. It does ubiquitinate under ST1. So it does something on the axis. So another particularity of this mutant, and why is it sticky, is because it's having a very, very, very strange ZIP1 cluster here. So here's the wild type, again, for comparison, with ZIP1 starting at the side of the nucleus, then half paired during zygotin, and then the chromosome are fully paired at packeting to dissolve at diploting. But in the mutant, you still have that, and then you have a very persistent cluster of ZIP1 protein, and it's still persisting, and eventually you will have synapses, but you still have this cluster for a very long time. And the other thing that the mutant does is having a lot of interlocks. So interlocks as when the chromosomes are not dissolved and they're basically locked together, and there's different configurations. So they can go above or under another. They can be trapped in between the two chromatids, and you need to have a lot of endonuclease work to resolve these things. This is normally resolved at the zygotin level, but when the synapsis is delayed, you can see it much faster. You can see it under the microscope, and it's not repaired in a fashionly manner. More importantly for us is that if you look at the chromosome axis, so here, for example, this is one chromosome perfectly paired, and on each of the thread you have like one or two, maybe a crossover site. And this time, we use an antibody gifted nicely by partners in IPK, Stefan Heckman. And then what it does here is high 10 is a pro-crossover factor. So we know that when high 10 is on the chromosomes, it normally makes a crossover, right? And when it's on the ZIP1 cluster, it's a lot, actually a lot of this foci, like a lot. And it probably looks like this. But then it makes us think that maybe that's the reason we have more crossing over in the mutant. And so we came up with a model, and the model is as it is. So the meiotic breakdowns and ubiquitin's role in releasing chromosome stalls. And here's what we think is happening. So if you remember, each of the parents need to be organized properly by the protein ASY1 first. So ASY1 come at the beginning of the process, start to load and organize the chromatin loop. So here is very disorganized, and then they will be very organized under the lateral element, which is all around the chromosomes. And at some point, the protein ZIP1, which is here in green, will be pairing up all along the chromosomes. And when the pair up is complete, then those chromatin loop, they are not inactive. This is where recombination has happened. This is where those loops start to cross over and to make a recombination. And this protein high 10, when it becomes very, very big with an agglomerate of high 10, it's securing the crossover, and that's lock. And that's it. Nothing else moves. There's no crossover nearby, right? Which we call interference. But in ST1, this is very delayed. It's not working well. Nevertheless, when it's working, you have very, very big concentrations of high 10 coming up. And so by definition, you can have one crossover, but you can have one crossover nearby, which means that we potentially remove this interference and potentially remove the issue that you have at the beginning of the talk, where you wanted to separate your desirable allele and your wanting your good allele. That's what we think. So that actually raises more questions than answer here, of course. So we're still not entirely sure how this ST1 work. We're not entirely sure how it shapes the chromatin, although we have evidence that it's not on histone methylations. What is regulating? We don't know which are the partners. We know it's ubiquitinating ASY1, but is that actually the real things? We're not entirely sure. Can we develop an inhibitor assay? So the reason we're saying this is because we think that ASY1 is not dissolved properly. And that means that will imply the proteasome pathway. And for this, there are probably inhibitors that we can try. But more importantly, can we release straight from Barley wild relative using ASY1? Well, that will be for another story. So here are some conclusions that I wanted to bring up. The first is Barley has a very tight control of crossover formation. And that is very difficult when you want to do conventional breedings. And that's the things, right? And so far, I think we now have quite a lot of resources that we can use for that. But we just need to have proof of concept experiment. That's just to say, right? Barley is a well-behaved eukaryote. Most of the time, things are happening in mouse and human and Arabidopsis, at least in meiosis, which is a very conserved mechanism, while you have the same in Barley, but we did see some differences. We saw differences between Arabidopsis. We even saw differences between human and Barley. And so my 2 pence forward here is if you want to look at a process in a certain organism, just do it directly, because that will save so much time. We knew about XRCC2 quite a long time ago, and I know a lot of people have tried, even though we told them it's probably a good idea. And yes, it is not a good idea. So we have found a novel gene in Barley. So that suggests that there's alternative pathway here for the control over combination. And to be fair here, we also have a lot of data showing that there's a lot of ubiquitin genes which are very dynamic in meiosis, at least at the transcription level. So that's something that we can explore as well. And so on. And there's an evident role of recombination in control of meiosis. And I think now Barley is adding to the system. And I think this is where we need to understand a bit more, because we don't know very much. So with that, I would like to thank collaborators and also everybody who worked on this project. Those mutants, like I said, there's like 15 years of like mutant work that we've done with Luke, Robbie, and everybody. Jamie and Isabelle have been extremely productive on ST1. They led the entire things, the absolutely phenomenal work that they've done here. And also we have a lot of collaborators from the University of Dundee, Birmingham, Leicester, Aberystwyth, and IPK. If you remember at the beginning of the talk, I didn't really speak about unlocking the centromeres, but that's because we've not done yet. But we have a plan. But the plan is to find a PhD student for this PhD, which has just opened. And the deadline is 3rd of April if somebody is interested. And thank you very much.
Thank you, Isabelle. Thanks for taking us through the dance. That's a really nice way of looking at meiosis, I think. I'm not quite sure how I can see the questions, but nothing can be right. Can I kick off with a question?
Mm-hmm.
You mentioned that meiosis is this incredibly complicated progression, tightly controlled genetically, that's the control is consistent right across the eukaryotes, from yeast right through to humans to plants, Arabidopsis and Barley. And yet you're saying you're picking up differences between the roles of some of these genes between Arabidopsis and Barley. Can you expand on that a bit?
Yes, I can expand. I can't tell you a final answer because I don't think anybody does yet, and I know there's a lot of studies. There's a lot of things that is being done to try to understand this process. But you need to remember that so Barley is a very large. The Barley chromosome are very large. I think that's the crux of the matter, right? So first of all, I think there is a genomic structure that plays into the game. So if I remember what you told me a lot in the past, there's a lot of chromosome rearrangement or within the during evolutions. There's inversion, there's ancestors, centromeres. All these things will affect how a chromosome recognize each other. That's the first thing, right? And the other aspect of this is also because it's a very large chromosomes. They also have, because they start by synapses start by the end of the chromosome, it's a much longer time to reach the centromeres and to pair them up. So if you screw up this pattern a little bit, the effect would be much more dramatic in a species with a large chromosome rather than in a smaller chromosome like Arabidopsis or rice. In fact, in rice, ST1 similar to rice is also not behaving the same, right? So that's another thing, right? And to come to ST1, this isn't grass specific. So grasses will have the unique way of doing things as well. So ST1 is one gene, but there are other genes that are different. There are other genes that maybe are not in an organism and another organism, and that's part of the evolution. So it's all this complexity that we need to address and things. For us, there's probably one thing that remains between all these organisms is that all the study that we've read so far and that we do so far seems to indicate that the remodeling of the axis, it's maybe where we can have the more flexibility in changing crossovers.
Okay. Thanks for that. If I look in the chat, I can see a question from Damping Lau. Would you like, I don't know if you can ask your question yourself, or I can read it out for you. No, it looks like I'm reading it. Basically, it's asking whether under the ST1 background, will performing many more crossovers cause chromosomal segments to be missing, or rearrangements, I guess?
I'm not sure. I'm not sure what is the question here. Can you repeat that? In the crossing.
With a ST1 mutant background, given there's potential for many more crossovers. Is there potential for chromosomal segments to become missing, or rearrangements, or are there structural issues potentially coming through that?
Yes, I think there will be. I think there must be, but I don't think that we've checked that at all. But the funny thing with these mutants is, so yes, there will be. Yes, there will be. There's probably rearrangement and probably loss and breakage and things. But I think what we saw as well is that when we look at those mutants, we have kind of a right. We have a lot of we pick up the myocytes which are not really looking good and things. So the majority look horrible, but there's a few that looks okay. And I think those few that looks okay, that's the one we picked up into the recombination data. So I think there's a little skewed here when we have the recombination data. What it means is whatever made it through is correct and have increased recombination and is useful. Whatever didn't make it through just dies somewhere else. So even though there's a lot of rearrangement, I don't think you would pick that up because you pick up only the good stuff. The cells are very, very smart on that. If something is broken, it's not going to go through. And we know that those stickiness sometimes remain at anaphase 2 in the second divisions. So those one, it's very unlikely that those one will go through the full division. They will just collapse. So I think I think there's a natural way here for the cell to select something which is fit enough for the next generation. But saying this, I know that in a background like MLH3, there's a lot of gene inversion and things like that going on, and so I think it would be very nice, cool study to do actually.
Okay, there's a potentially related question from Andrew Lloyd saying, is he right that
Hi, Andrew.
you see incomplete synapses in the ST1 mutants, but a global increase in crossovers? Or do you get complete synapses, but is it just delayed?
It's complete synapses, but it's just delayed. And sometimes it will retain this telomere agglomeration for a very long time. So synapses will happen. And Andrew, I know you're familiar with this, but it's very much the story of it has been developed by Chris Morgans some time ago, where this coalescing model where partners like high-10 were coming on the chromosomes, and then they kind of congregate around the potential hotspot of crossover. And then when they are big enough, we're not entirely sure what is the mechanism for that, then it stops. Everything else is removed from the chromatin, and then only the crossover remain. And I think this is a bit of what is happening here. This is a bit of what's happening. What stopped the signal, I'm not sure. We've not, I'm not sure even how to look at it. But yeah, if you want to do some mathematical model on it, I'll be very happy, because honestly, that really breaks my head sometimes. So,
I mean, could I ask a related question? In when you have these clusters of appears zip stickiness, if you like, is there associated clustering of ASY1 as well? Is the condensation of the chromosomes affected, as well as the? I mean, is the axis abnormal, if you see what I mean?
So they look normal. For but for an expert with the microscopy image, they're actually not entirely normal. Now the problem is with fluorescent microscopy, and especially with the confocal, or even with the structural elimination, I can't see how I could accurately measure the quantity of chromatin access. I really can't see it. I can't see how to do this. And so my next to-do list is, I've got some plant growing, and I'm going to try to see if I can see better with a STEAD imaging, which is a much more deep foci focus and things that we can do. The other thing I want to do, and that's a lot more accurate, will be if we could do a chip sequencing with ASY1, because then you can see, you can quantify it. You can quantify it, because I got the feeling that if my model is right, then I think that ST1 doesn't have the turnover of ASY1 that we normally expect. So the theory here is ASY1 comes the access, but when ZIP1 needs to come up, some of the ASY1 come out, they are dissolved, they are destroyed, and then they come up again. So basically, there's a high turnover of those protein on the axis. But we think that this is delay in the mutant, and this is why you get ZIP1 that cannot actually do a proper elongations. So that's a problem, right? So here it's again a delay mechanism, and we know it's delay because we had a lot of data showing that when it should be normally at packaging is like zygotine or something. So I think for me to answer that, we'll need chip sequencing to be accurate. But there is a higher intensity of signal of ASY1 in all my images, which will be consistent with a different organization of that protein on the axis. Yeah.
Yeah, thanks. There's also a follow-on comment from Andrew Lloyd. Perhaps the interlocks are inhibiting the coarsening of high-10. Could you comment on that?
Yes, that's totally possible. And the question is why the interlocks are there. And that's another question, right?
So
and I
why
think,
are the interlocks there?
Yeah, what are the interlocks, right? And I think again, they're interlocks because they're not getting repaired in a fashioned manner. And I think that's because the axes are not properly formed, right? So even though we see a nice green colors and a linear chromosome, I don't think that is actually accurate. I don't think, and I really believe that is not entirely properly done. But the microscopy image that I have just now, it can't show that. You know, it can't show that. I can't quantify it. So again, I think again the chip sequencing, but also if you stretch the system a bit. So for example, if you were to play with a protease inhibitor, do we reproduce this phenotype? You know, do we get the same phenotype? You know, for me, the best will be if I could find somebody to help me to navigate this AlphaFold three-dimensional protein where we can maybe predict at the protein what kind of inhibitor we can develop, and that would be great because we can try to inhibit the protein rather than inhibit the whole system with protein inhibitor and, you know, stop every other E3 ligase. But yeah, there's a lot of good things to do here actually.
Just check if there's other questions here. If not, may I was probably again ask another question about ST1.
Yes.
You mentioned there was a stickiness in anaphase 2. That implies there's a role for SY1 later on in meiosis. Is there any known functionality of the protein later on as well, beyond protease?
Haha. Functionality of the protein, I'm not aware of anything yet, but we've published in 2017 about that. We've published the what I call the tinsel paper. So if you remember about the tinsel paper, is when you have to desynapse. So I didn't talk about desynapsis today, but this is also an interesting study here. So when synapses needs to be dissolved in order to prepare for metaphase, the SY1 axis kind of making stretches and loop around the chromatin area. And I think it's a way to get out of the axis to open up the chromosome and then to prepare for separation, right? How it does that, again, I'm not entirely sure, but I wouldn't be surprised if the proteasome pathway is not involved in it, because in her mutant is a little bit compromised as well, and in Meiotic it's even worse. So there is something that you need to have a proper synapsis as well to have it. But then I've been silly enough to actually look at what will be the labeling of SY1 later on in meiosis. So in fact, I looked at metaphase, couldn't see anything. But then I saw at anaphase 1, before anaphase 1, and what it does is there's a green signal, which is like SY1-like signal, but it doesn't do anything like an axis. What it does is to be around the chromosome, and it seems to be pushing them away from each side of the nucleus. And in fact, talking to Stefan some time ago, he's developed some line for SY1 in Arabidopsis for a long time ago, and he told me that what he sees is that SY1 is pushing from one side to another, is going to one side or the other of the cell very, very rapidly. And that is also a similar mechanism that you see in some one of the yeast, where you get this really, really back and forth protein that help the chromosome to push towards. So there may be something here, because we see the signal in metaphase 2, anaphase 1, but also in anaphase 2 and even in the tetrad. And in fact, I remember when I visited the lab in Czech Republic from Yaroslav at some point, we did some flow sorting on here. And we were flow sorting chromosome that are cells that was fixed and labeled in solution with the protein SY1. And it was like, this is funny, because I could also see the signal later on in those later state of meiosis. So I don't think I'm making up. I think it exists, and I do think that there's a role here, but I don't think anybody has explored that yet. But maybe I'm wrong. But yeah, there's something.
Oh. Plenty yet to find out.
Yes.
I realize we're coming up on the hour now. If there's any more questions, the hands up. I think we might have exhausted.
Dancing is exhausting, isn't it?
It certainly is. Okay, I think I'll wrap it up there. Thanks again, Isabelle. That was
Thank you very much.
A really nice taking us through the initial Kaylee of the dance. So thanks again, and please look out for the next seminar on your inbox. Cheers.
Thank you very much. Bye, everyone.